My name is Whittaker. I'm the Director of Pharmaceutical Safety Screening at Vala Sciences. My background's in cardiac physiology and cardiac disease. I joined Vala about 5 years ago. I developed a number of assays for them early on while they were completing the KICK prototype, which is our kinetic image cytometry platform that we use for our arrhythmogenic liability assay. Vala's core technologies are really built around advanced high-content or imaging-based applications. And we span the range of discovery from very early discovery with our ultra-high throughput screening platforms, which can be used for primary screens, through to our kinetic image cytometer, which is a high-throughput cell-by-cell analysis of live cells. With the KICK technology, what we're able to do is take the whole field and identify each individual cell within the field of view that we're looking at. And then we can do an analysis on what's happening with each individual cell. And that gives us a really detailed way to get into the functional biology that's happening. When you look at some of the other products that are out there, they have an integrated approach. So if there's any kind of heterogeneity within the cell culture, it's missed because you're doing this integrated approach and it's masked. So we find that this cell-by-cell approach is actually a little bit more detailed. We're using the calcium transient as an integrated signal. So calcium transients are the second messenger that takes the electrochemical information from the action potential and turns it into contraction. Since we're looking at intact cardiomyocytes and intact human cardiomyocytes, in the case of the eye-cell cardiomyocytes, we can also ask questions about signaling, effects of the compound, and trafficking effects, all of which can relate to or cause changes in cardiac excitability that may potentially lead to the development of arrhythmias, which is of a major concern. And also with these cells, we can actually see the arrhythmias in the dish because we see things like EADs, which are single cell phenomena, and we can pick out individual cells which are throwing EADs, whereas the rest of the culture may not be. With the eye-cell cardiomyocytes, since it's a scalable model that's reproducible, the effects are reproducible in the cells, and we can rely on each vial to behave a similar way, that really enables us to deploy this as a high throughput screen as opposed to a screen that we're doing with primary cells. You really can ask a lot more questions with an intact cardiomyocyte model with a functional readout than you can by trying to take the reductionist approach and look at ion channels individually. It's kind of a perfect model for doing this sort of screening. We've probably had about 120 compounds that we've screened through our assay with the eye-cell cardiomyocytes. And we see a very high level of predictivity, greater than 90% for flagging compounds. And then when we go in and we actually look at the sensitivity of the assay and compare it to where you would see these effects in the clinic, it's very, very accurate as well. So we've been working with Cellular Dynamics for a little over 4 years now, and it's been a great partnership. It's really helped us speed along development of the assay using the eye-cell cardiomyocytes. And I think from CDI's side, they've been very supportive of technology developers, which is very helpful for a small company like us. And at the same time, we've been able to learn a lot about their cells and pass that information back. There's a huge amount of data around these cells that we can pull from and that you guys have to work with, and I think that's a great relationship.